And so with that, let me just say that it is a pleasure to be here today. My name is Rusty Poole. I'm in San Antonio, Texas. I'm an IVF lab director, scientific director, Fertility Center of San Antonio. Very pleased that you're all with us. I want to quickly acknowledge the number of people in the background of the IVF, in the International IVF Initiative, the platform that has been so good in helping us to remain in contact over these rough times. But we have in the background some fantastic people doing wonderful things for the computer world. We have Thomas Elliott and Marianne from IVF Meetings, but we also have scientists that are helping in the background that'll be ferreting questions from you. So I want to give a quick thanks to Brian Lamonto, to Shaistu Sedrudin, to Anna McLaughlin, Peter Naj, and Jacques Cohen is also in the background somewhere, and Giles Palmer will be helping out as well. Thank you very much for all of your help. I want to take this opportunity to tell you that we have a fantastic cadre of speakers, 3 international experts in this field, but we actually have a 4th as well. And I want to introduce my co-moderator, Dr. Gary Smith. Gary is well known to you for his current work in stem cell technology and his work in bringing microfluidic technology and engineering into the art world, but he also has a passion for the field we're talking about today. And in his career, he's done quite a few things to look at the enzymatic regulatory machinery that remodels chromatin and brings about oocyte maturation, and he's quite the expert. So we have 4 experts. Gary got his PhD at Washington State University. I met him, oh gosh, probably 25 years ago when he was a postdoctoral fellow finishing up under Professor Don Wolf at the Oregon Regional Primate Center. He took his first faculty position at University of Chicago, but eventually was recruited to his current position, University of Michigan. He is a professor of OB-GYN, a professor of molecular and integrative physiology, and a professor of urology. And Gary, take it over, and what are your thoughts on today's meeting?
Great. Well, thank you very much for that introduction, Rusty, and thank the organizers for letting me be here with you guys today. I think it's very exciting to see and to listen to our upcoming speakers. And you know, this is something, an area that I know many of us have felt very deeply about, is that it's not just sperm and embryos, but it's the oocyte too. And it's really that background of the oocyte that, you know, doesn't drive the whole developmental process, but actually does have a very, very significant contribution. And I think that's going to be very, very nicely talked about today by our 3 speakers. So if you want to go ahead and introduce the first speaker, we can go ahead and get started.
I will do that. Well, thanks again, Gary. And just a quick word about what initiated this session. It's just, to me, one of the most striking things over the 38 years I've been at ART is the gulf that's developed and actually has widened between our fundamental knowledge of oocyte biology and then our day-to-day application in clinical practice. We seem to be going down the rabbit hole of more and more work, and we quickly gloss over this important biology. And it just seems to me that we've gotten into this era where the presence of a polar body just says we're good to go, when in reality, nothing could be further from the truth. So we have precipitated this session, and I'm very pleased to introduce our first speaker, Dr. David Albertini. David is a PhD product of Harvard University, and he did his postdoctoral work at the University of Connecticut, but he returned for an academic appointment at Harvard. But since that time, he's had meaningful chairmanships and leadership positions at Tufts University in Boston, their School of Medicine. For years, he was with the University of Kansas Medical School in Kansas City. He has worked for the Center for Human Reproduction, but he's now returned to New England in his current post. So he is a professor of reproductive cell biology at the Bedford Research Foundation in Massachusetts. And as most of you know, he is the guiding light that has brought the Journal of Assisted Reproduction and Technology into really the realm that it occupies today as the editor-in-chief. He's done such a stellar version of guiding the development of that journal. So with no further ado, I want to introduce David, and his topic is how oocyte maturation regulates the human embryo's developmental competence. So David.
Thank you, Rusty. Thank you, Gary, and also to the organizers. It's a pleasure to be participating in this effort, and I think, as Rusty has just said, we're going to be trying to fill a gulf here between the world of clinical biology and the basic biology of the oocyte. Okay, good to go. So I have one disclosure, as was just mentioned, based on editing jar. And what I'd really like to do today is get into a dialogue, okay? A dialogue that is between the physicians that we all work with and those of us in the embryology lab with regard to what the quality of an oocyte is, what dictates it in particular. As I'm going to point out here, we know that the clinical directive behind ART programs is to manipulate the hypothalamic pituitary ovary axis in the practice of controlled ovarian stimulation. And you know, this is the dialogue, of course, that our IVF centers have with our patients who are anxiously awaiting to know, well, how many follicles have grown, and what is this going to look like in terms of the number of oocytes retrieved and my chances of having a baby. This is the overarching question that we deal with. And I'd like to argue with you today that the dialogue on a much more practical and fundamental biological level is really about one between the follicle as an endocrine entity and the oocyte as a paracrine force within the ovary. My take-home message is that it's time to look for alternatives to traditional controlled ovarian stimulation, because in its current form, the conversation that's being carried out between physicians and their patients has much to do with driving the development of the follicle, but it leaves the oocyte behind. So why are we having this conversation? I think we all realize that a very small fraction of the human eggs that are retrieved after controlled ovarian stimulation make babies. We know some don't mature, some fail to fertilize, many arrest before they get to blast or potential transfer, and even those that we rank highly and go on to transfer, many do not implant or certainly make it to term. And what we suspect is that among the many factors in play that will determine whether a given oocyte can produce a term pregnancy, one of the things we suspect is that COS may be altering in unknown ways as of yet the cytoplasmic maturation or the cytoplasmic quality of the oocyte that is achieved after a triggering stimulus for ovulation. And my purpose today is to try and connect this problem of cytoplasmic maturation, look at it in a more sophisticated way, and ask, is what we know at this point encouraging enough to prompt development of other strategies for collecting oocytes and ARTs, or in adopting in vitro maturation? So I'm going to provide you with a bit of an overview of what the biological problem is with cytoplasmic maturity and the clinical problem. I'm then going to ask my colleague Gio to take a look at that transition from the meiotic world to the mitotic world, from the events around fertilization and maturation to the earliest cleavages, as he and his colleagues have shed new light on the dynamics, the biological dynamics of what it is about an oocyte that links to the competency of the embryo. And finally, I'm going to ask Rebecca to try to put some of this biology into the perspective of what we know about in vitro maturation now, and just how close are we to adopting this technology for the safety of both mother and baby. So this is a journey, and it's a road that we're going to take from first addressing the biological problem of cytoplasmic maturity, and then moving on to the clinical problem. And really, the biological problem has to do with a very long history of oogenesis, but for our conversation today, we're going to combine it. Here I can use my pointer over here. We're going to confine our conversation to those events that occur after primordial follicle activation that sustain what we used to refer to as the gonadotropin-independent phase of follicle development, and then the gonadotropin phase, which is what, of course, we drive within each new cycle that we initiate for a patient. Professor Goujon years ago suggested that this was a process that took about 100 days in the human. And along the course of follicular development, it was expected that the oocyte would gain the competence to perform a number of functions. And we're going to define those in a minute. But for today, the question we're going to begin to ask is, what is it in that oocyte that it acquires during its ovarian journey that will allow it to make a blastocyst and, if transferred, make a baby? And bear in mind that we are indeed a monovular species. We are not used to performing like rabbits or other rodents and rats. So we normally dwell on the selection of the dominant follicle, which of course is where the clinical problem comes in, because it is the design of controlled ovarian stimulation to bring as many follicles to fruition as possible in order to make as many embryos as possible. And I'm going to leave you with this slide as really the basic design principle that we're dealing with here, which is the competence of an oocyte to support all of maturation and the events up to implantation is a reflection of the metabolic support that the follicle provides that oocyte with. And there are manifestations in the physiology of the follicle, which has really become enriched in terms of our knowledge about what drives the development of the follicle, because it's designed to both serve and protect the embryo from genetic or epigenetic damage that can happen after fertilization. And the question then becomes, does controlled ovarian stimulation, or for that matter, even our principle of extended culture, impact the inherent capacity of the oocyte to produce a high quality blast? Well, the biological problem is very complicated here in 2020. We have come to understand that it not only encompasses a long period of time, some 3 months worth of time, most likely, but it also engages a very complex series of interactions that have little, if anything, to do with the steroidogenic properties of the follicle. Indirectly, gonadotropins play into these schemes, but we're now shifting our thinking about what it will take to yield oocytes that could sustain development. And so this slide just summarizes for you 2 of the main areas that we've come to understand. The first is that there is an elaborate communication system between the oocyte and the surrounding somatic cells of the follicle that's founded in these paracrine factors, BMP15 and GDF9. And moreover, over the course of folliculogenesis, the types of signaling interactions between the germline and soma change. In some cases, these are subject to gonadotropin modulation. It's not only a complicated process, but how we view maturation has changed dramatically over the last decade. We're all, as Rusty just referred to, used to noting a polar body and basically concluding that here we have a mature oocyte that's ready for IVF or ICSI, and we're going to launch it on its merry way down embryogenesis. Okay? While nuclear maturation, we've understood for years to be a fundamental problem in cell cycle control and the cytoskeleton, but it doesn't stand alone. It's coupled with many other processes that are going to go on during maturation that will dictate events in embryogenesis. So let's just look at a few of these. Cytoplasmic maturation in today's vernacular really involves 4 interrelated processes. There are the metabolic modifications that take place in the ovulatory follicle subsequent to the LH surge. Okay, and these are cumulus cell dependent And they essentially load the oocyte with not only bioenergetic substrates, but other important molecules like reduced glutathione, which are necessary for male pronucleus formation. In other words, the bioenergetics and the translational control of early development, despite what you've heard about embryonic gene activation, are largely controlled by the oocyte as a result of metabolic modifications made during ovulation. That's not all. We've come to recognize for years that the perifertilization biology, migration of the cortical granules into a fusogenic position, loading of calcium stores to establish spikes that will support egg activation, etc. These are events that occur in the cytoplasm during that time. There's now abundant evidence that not only primary epigenetic modifications in terms of DNA methylation, but the expression and storage of enzymes that will be required for subsequent epigenetic modifications in the morula are all functionally organized during maturation. And finally, there's a fairly new described process called cortical maturation that involves tyrosine kinase signaling pathways that mediate remodeling of the actin cytoskeleton that have been shown to be directly related to the incidence of fragmentation in the embryo. So our thinking is broadening as to what cytoplasmic maturation is, and at least this gives us a handle for what we need to be focusing on in the future. So we can begin to ask, if the oocyte is endowed with these properties as it matures, why is it that so many embryos fail? And we're going to actually do the balance sheet a bit later in the clinical section on how many embryos fail that are made from the oocytes we retrieve. And in fact, it should not surprise you that between cell cycle defects, metabolic insufficiencies, protein homeostasis, which is a big one now, that these are all properties endowed in the oocyte that will dictate very specific competencies after fertilization, some of which are listed in this table on the right. We tend to forget that the zygote inherits all of the machinery needed for pre-implantation development, not just the mitochondria, ATP, or ribosomes, all of the precursors for the Golgi complex for every single cell come along. This is the true mother lode, and I think we've lost sight of just how important these things are. Other principles that have come to the forefront in the clinical evaluation of human embryos that lack in their developmental potential have to do, of course, with genetic integrity. We've seen very much, much work now in the days of PGT to emphasize, if not overemphasize, the genetic liabilities that the human embryo takes. And depending on the nature of those, embryo arrest is a common consequence of that. That there are, now known disturbances in mitochondrial function. Again, it's probably one of the bigger ones that's gathered a lot of attention recently, has to do with how do you preserve and protect the integrity of the genome, protected from DNA damage? Are the repair mechanisms there, not only for the female genome, but for the paternal genome, to ensure that the genome arrives at the time of implantation in a reasonably healthy state? And again, I think it's important for us to keep in mind that all of this capability or competence is inherited from the oocyte, except the sperm centrosome. I could walk you through some of this data, but it's very well recognized now. This is just one example coming from the Albertini and Handyside group about the nature of the defects in cleavage that have been ascribed to genetic abnormalities, specifically aneuploidies of various kinds. These are common in human embryos. And as you'll hear, sometimes these are rectified or corrected. Other times they fail and the embryo ceases to develop. So let's begin to shift now. That's the biology, okay? Let's begin to shift to the clinical problem. And at the heart of the clinical problem, of course, has been the various types of ovarian stimulation that have been adopted over the years. And for example, the widespread use of human chorionic gonadotropin as an ovulatory trigger. We'll come back to that at the end. But these are all subjects that continue to be actively investigated in the human with respect to trying to improve pregnancy rates and trying to improve outcomes. So the clinical problem really reduces itself to a matter of how we are manipulating the physiology of human reproduction and specifically ovulation, because that is our window when the maturity of the oocyte is achieved. It has been driven by a desire to collect as many oocytes as possible. I don't think anyone would deny that. And the question we can begin asking today, of all those oocytes that are retrieved and are from a good responder, a poor responder, a donor, an older patient, okay? Of those that are retrieved, how many of the oocytes are viable? How many are mature? The state event metaphase 2, when we collect them, fertilize normally, etc. How many actually make babies? This has been a very contentious subject. And I think, as most of you know who work in the embryology lab, this is the hard reality that we face on a daily basis in interacting with our physicians and patients. Well, let's go back to basics for a moment. There are principles of follicular physiology that were based largely on the so-called two-cell model and two gonadotropins, which we now know is a serious oversimplification of the physiology of the Graafian follicle. But historically, this area has developed as a means for sustaining folliculogenesis at the expense of oogenesis, is what we're now coming to understand. And this can no longer be viewed, if you will, in a follicular-centric way. We have to turn our attention to the oocyte and the changes it will undergo during the course of maturation to ensure its developmental competence. Another thing that complicates matters is over the last 3 decades, we've seen a wide range of controlled ovarian stimulation protocols adopted, tested, and discarded. We've gone lately back to using GnRH agonist or antagonist, so using a patient's own releasable quantum of gonadotropins. To, of course, the many exogenous versions, okay? I think hopes were very high when the recombinant products initially came on the market. We've got a wide range of patients who respond in various ways. Our monitors, inferences, if you will, for what the quality of the oocyte will be are what? They're all markers of follicle quality. None of our typical monitoring markers directly reflect the quality of the egg. And, finally, we have to remember that by its very nature, controlled ovarian stimulation manipulates the natural luteinization potential of the Graafian follicle and supplements, etc. And that we are in the middle of changes over these last 2 decades is very well illustrated by this paper that recently came out from the Danish group representing the Nordic study. And they again remind us of the variables within which we are trying to make some sense out of this problem of cytoplasmic maturity for the benefit of our patients. And so it's not just a matter of managing and micromanaging the menstrual cycle, whether you're using urinary products, recombinant products, GnRH antagonists, but we'll come back to this, the trigger. What is the dominant trigger we've used over these years? We now are able to move to single embryo transfer, and we have progressively increased the time in culture for the embryos. When we reflect back on this in 10 years, we're going to need to ask how the conditions that the oocytes were allowed to develop in influence this final ability to make a term pregnancy. So coming back to the question, just how many of the oocytes that we retrieve ever result in a baby? And as many of you know, Pasquale Patrizio and Denny Sackis approached this problem a number of years ago. And in their initial papers published in 2009, where they compared essentially the number of oocytes retrieved with the usable embryos relative to baby output, essentially emphasized a point well taken about maternal age. That in patients over 35, something less than 5% of all the retrieved oocytes would result in a live birth. And in patients under 35, it was more. Well, these were pretty crude initial studies. And so now they and others have been able to go back and say, well, what about all of the oocytes that we get to go to cleavage stage? What fraction of those will make a baby when transferred? And as you might again indicate, 30 to 40% make babies if patients are under 35, whereas that drops to more like 10%. Now we have, with the increase in the use of donors, we can look at the population of donors on a more pristine landscape and address this question even further and look for what I would call a best case scenario. And what that comes down to is, again, something on the order of 10 to 12% for good responding oocyte donors. What does this mean? It means absolutely that there's still a very small percentage of oocytes we retrieve that have the ability to establish and maintain a term pregnancy. And from our best estimates, which have been recently recalculated by Wilcox and colleagues, we know that people, fertile couples, undergoing timed copulation and timed ovulations, best case scenario in our species is 33% of all conceptions will go to term. So COS and ART practices, partly because they're a moving target, still result in oocytes being retrieved and used to make embryos that lack the ability to take the problem to term. Okay, I'm just going to move here and try to summarize the main points of what I discussed. I did just gloss over the situation of HCG in particular as a trigger. There's some interesting work done historically in animals and more currently in humans, and we can return to that point in the question and answer if you would like. What the sum data at this point is telling us is that controlled ovarian stimulation interrupts the process of oogenesis. And it does so by preventing the reciprocal control of FSH and LH responses that lead to an environment in which the oocyte cannot fully mature, number one, and number two, an environment, as we know, that is subject to premature luteinization. These precocious increases in intrafollicular progesterone have been shown to impede the acquisition of not only meiotic components, but mitotic components in the embryo, and they do so by destabilizing the oocyte cortex and increasing the risk of fragmentation and cytokinesis failure. You'll see some examples of this in the next talk. This suggests that the way forward may be to reduce both the levels and extents of follicular phase stimulation, combined with IVM, as you'll hear from Rebecca, under controlled situations where the dimensions of cytoplasmic quality that we know are so essential for embryogenesis can be reproducibly achieved. What does the future hold? As I just suggested, maybe less stim and more IVM? I don't know. But we have to accept the fact the oocyte we now recognize to be in charge of follicle development. It is not the other way around as we previously thought. And at least up to the time that the follicle acquires its sensitivity to LH and FSH and becomes an endocrine organ to meet the demands of the conceptus endometrium interactions, we need to pay closer attention to that dialogue between the follicle cells and the oocyte, because in the end, that's what will count the most for embryonic success. In fact, if you take a broad look at this problem, the biological problem, it's clear that the oocyte can take care of its own business, at least in terms of bioenergetics during maturation and those early stages of development, as long as it is properly nourished while it was in the follicle. There are some metabolic pathways that have recently been resolved to show this to be a unique property of the mammalian oocyte, and one that can sustain the bioenergetics up to the time of blastocyst formation without consumption of glucose. How ovulation is induced matters, and it will have consequences on the expression of maturity as we work through this, not so much with respect to the output of the follicle, but with respect to the quality of the oocyte. So finally, let's think of maturity in this way, in the sort of as it's done in the financial world. But maturity refers to a finite time period at the end of which the financial instrument, the oocyte, read 100 days, will cease to exist. And the principal, the embryo, is repaid with interest. That is, if it finally implants properly and leads to a term pregnancy. So I'm going to close there, and thank you very much for your attention.
Well, great. David, thank you so much for reviewing that. That was a great review. We do have a few questions that we're going to go ahead and start with, and then go on to our next speaker. So, you know, David, I think one of the nice things that you pulled together here is really the importance of the link of cytoplasmic maturation and nuclear. You know, we know they're different. We know sometimes nuclear can happen, cytoplasmic doesn't. The definition of that has been, I would say, problematic over the years. But Mike Reed has a question here, and I kind of had the same question. As you look to the future, you know, what might be those live cell biomarkers, whether they be oocyte or follicular or cumulus cells, but something that can give us a clue as to what is the developmental competence of the oocyte? Because I think, just like you're saying, looking for just a polar body only or anything we see at the light microscope level has not really allowed us to go beyond the first steps.
Yes. Wouldn't we like to know? The biomarkers for oocyte quality. I won't go into it, but that was a conversation I had with Howard Jones many, many years ago. I think the sad reality at this point, Gary, is that there are no tests that I know of that are direct reflections of the developmental competence of a human oocyte, okay? You know, metabolomics, I think, is a very active area, but again, You know, how directly is that going to be related to all these other properties that I've just summarized that have to go on for the zygote to complete its journey? So I personally, I don't know of any tests that really do a good job in a predictive way of monitoring this. You know, one of the big things has been, you know, it has to be non-invasive, okay? It has to be non-perturbing in many ways. There are, as the audience may know, there are some techniques being developed with mouse eggs now using the endogenous fluorescence of important substrates like NADH. You know, maybe these will open up some channels for doing this. Again, you know, the concern all along for those of us like you and I that have been using fluorescent markers over the years, you know, has always been, you know, the bad rap that you're going to take, because the dyes, once you excite them, are oxidatively potent. Okay.
Right. Well, and I think your point is very well taken, is that, you know, and this I think happens, we've all wanted to have some marker, but I think as technologies develop and we have more things like single cell RNA-seq and single cell proteomics, it really does open up that opportunity for us to begin to ask with live cell, not imaging per se, but live cell assays, and then future developmental potential, what might be a biomarker. It's going to take a lot of work, but now we're starting to get some of those tools available.
Yeah. And one of the things that you mentioned is, you know, what about, and I think people have hoped for a long time that there could be sort of biomarker patterns that could be tracked from the cumulus cells that would be reflective of, you know, the quality of the oocyte, okay? You know, and there too, with the sensitivity of the tests that are coming out, it might be, I think it'll probably be some sort of multi-parametric. It'll be, you know, it'll be a look at the whole picture from the follicle. It won't just be the oocyte secretome or the, you know, the RNA-seq from the cumulus cells. But let's wait. It's going to be an exciting time.
Well, I'm looking at the screen here and I see a lot more questions, but we're going to move on to our next speaker. Obviously, at the very end, we're going to come back and we'll have some more questions. But thank you again, David, for that first discussion. Our next speaker is going to be Professor Giovanni Coticchio. Giovanni is from Italy. I have had the honor and privilege of knowing Giovanni, and many of us have for many, many decades. Done some stellar work in the area of oocyte cryobiology, also in the area of just basic understanding of what happens to the oocyte during cryopreservation, during in vitro maturation. What are some of the markers? And so it's really one of the great things in this field is that we've been able to get to know each other, challenge each other sometimes, and have some great discussions. Giovanni is a scientific director of 9 Baby Family and Fertility Center in Italy. He has a PhD in cell and developmental biology, 80 plus papers and peer review articles, a couple book chapters. Giovanni, like I said, is somebody that is always open to talking with people about his passion and answering their questions. And today he's going to talk to us about the continuum between oocyte maturation and early development. David set it up really nicely, and I think I look forward to seeing Giovanni's talk. So Giovanni, are you able to come on and share your screen?
Yeah, thank you very much, Gary. And I have to say, I was inspired by many of you, so I should thank you for being here at this stage. So I'm in the process now to share my screen. It should happen in a second. I think you can now see my presentation, my first slide, which reports the topic of the presentation, which is the continuum between oocyte maturation and early development, which is per se a very fascinating topic. So for the exercise of this talk, necessarily I needed to restrict the number of items to discuss. And I chose only a few of those very nicely introduced by David Albertini. So I decided to focus on two major themes, which are the importance of this cytoskeletal integrity in terms of cortical actin, and with respect to the process of polar body extrusion on meiosis 1 and 2, as well as spindle integrity. But there is also another major topic I'd like to discuss, which is the redesign which the oocyte undergo during transition to become a zygote. And this is strictly required for assembly the two parental genomes. So why is the integrity and asymmetry of oocyte cytoskeleton so crucial for development? So let's start with cortical actin. We want cortical actin to be interrupted. We find a meshwork of this cytoskeletal component just beneath the oolemma. It has to be intact to show cell integrity and therefore some aspect of oocyte quality. And at the same time, we want to have a polarized cortical distribution of actin, because this is very fundamental to give different cortical tension to different areas of the oocyte cortex, and therefore assist extrusion of polar body 1 and 2. We also would like to see, ideally, in a mature oocyte, as we know, even too well, an asymmetrically positioned and intact meiotic spindle to show fidelity of chromosome segregation and meiosis 1 and 2. And we also know that this beautiful structure is very sensitive to both intrinsic and extrinsic factors. So I have some data to share with you in a little bit more practical terms, which concern, in the first place, the cortical actin. So in a mature morphological normal egg, what we would like to see is an uninterrupted, continuous, in other words, ring of actin in an optical section of confocal microscopy of this cell. Of course, in 3D, you would see a sphere of meshwork of actin. And this is found in virtually all morphologically normal oocyte. And we can even to easily appreciate how this is important to assure for the cell to have his own integrity in terms of shapes, in terms of for assisting the ensuing division at the first steps of development. But in some cases, we observe that this ring, bidimensional speaking, of actin is interrupted, has lost integrity. And the important thing to remember here is that there are some subtypes of morphological abnormality of the oocyte, which are represented by accumulation of smooth endoplasmic reticulum and cytoplasmic granularity, which are strongly associated with an interrupted actin beneath the cortex. And I can infer, and I trust you can do that the same together with me, that there are implications for integrity of embryo cytokinesis, especially at the first mitotic division. Another aspect of the cortical actin to appreciate is that during maturation, and here we are towards the end of the entire genetic process, the actin undergoes polarization. In other words, at the GV stage, as you can see here in these two pictures, the cortical actin is uniformly distributed throughout the area just beneath the oolemma. But as soon as the spindle is formed, there is an accumulation of actin just in the area and somehow adjacent to the spindle. So we have an oocyte with a polarized actin structure, which is suggestive, in fact, of different mechanical tension of different areas of the oocyte surface. And again, this may have a developmental implication. One of those could be defective extrusion of the first polar body. I'm sure that it is an ordinary, although not very frequent, experience of embryologists to see these oocytes with very large polar bodies. And my suggestion is that that could derive from loss of altered cortical tension at the site of extrusion of the first polar body. And if we look more carefully at micrograph of this oocyte with large polar bodies, we can appreciate how not only is the polar body affected, which is large, and it is a bad news because the oocyte loses material that can be used to build up the embryo constitution, but at the same time, the meiotic spindle is largely disturbed. And virtually in all of these oocyte with large polar bodies, we have disrupted spindles, in both in terms of structure and also in terms of localization. For example, here you can see a spindle which is far away from the cortex. But instability due to presumably disturbed cortical distribution can also affect meiosis too. So we are already at the fertilization stage, and there's a very critical transition stage between oocyte and embryo life. So I'm going to show you a video that will start with a sequence taken more than 10 hours post-insemination. So you can see here what is supposed to be a nicely normally fertilized egg with 2 pronuclei. And I'm going to show you how the second polar body shown here becomes unstable and is reabsorbed by the oocyte, by the egg, I should say, at this stage, and a third pronucleus is formed, as you can appreciate. So these are implications, of course, for human embryology, because it gives us an idea of the potential of time-lapse macroscopy, but also suggests how time-lapse macroscopy per se can give a very potent support for understanding human development, irrespective of clinical treatment. And fertilization, once the life of the oocyte has come to an end, is a very critical time, probably as critical as gastrulation or maybe marriage, I don't know. So what you see here is between oocyte and egg, a time, actually a period, a short period of say 24 hours, during which the same cells has to reconvert much of its own array of organelles and molecules to move from a meiotic modality of division to a mitotic one. And there are differences. If you look at these cow spindles of oocyte and zygotes, you can already, just by naked eye, naked so as to speak, how the 2 spindles are very different in shape and size. And for the spindle, this structure is a manifestation of function. But let's just take a look at the meiotic spindle. The spindle can be studied nowadays in many ways. We can take very many measures and study in many different ways. Over the years, we have learned how it is affected by intrinsic factors such as female age, or by manipulation, for example, cryopreservation. For the sake of this presentation and also for the one that will follow, I also would like to mention the fact that when oocyte matured either in vivo or in vitro are compared, and when I say IVM, I mean the real IVM, not leftovers from stimulated cycles. The spindles of the 2 types of oocytes are virtually indistinguishable and therefore entirely comparable when they are assessed for more than 16 morphometric parameters, including major axis, minor axis area, microtubule density, etc., etc., etc. So the inference here is that unlike the mouse, at least for IVM, the spindle is able to develop normally during IVM. But the story is not over. Once the initial stages of fertilization are achieved, there is another critical step, which is the very first cleavage division. And again, time-lapse microscopy has come to play a major role in clinical embryology and has revealed us a phenomenon which was barely imagined, but now well documented, according to which at least 14% of all embryos undergo direct division. All zygotes, normal zygotes, undergo division into 3 different blastomeres at the same time, which is a very large fraction. The bad news is that, understandably, is that implantation rate is negligible, more or less 1%, and I'm not even sure that we can achieve term pregnancies with those embryos. So some studies have been carried out for the possible reason there's a very abnormal behavior, and a reasonable hypothesis is that this trichotomous division derives from the formation of a tripolar spindle, clearly a largely very abnormal organization of the mitotic spindle. In this fashion, of course, depending on how chromosomes, actually chromatids, are segregated in the sister blastomeres, we can have, for each couple of chromosome, either maternal loss, paternal loss, or even nullisomy, with disastrous consequences for the embryo. And digging even more in the possible causes of this situation, it's important to notice the case of the genetic locus polokinase K4, which is a genetic locus encoding for a product which is accumulated during oogenesis. So a truly maternal derived factor. We know that alter expression of this locus is associated with centrosome amplification and therefore formation of multipolar spindles in cell lines in vitro. And very importantly, maternal, but critically not paternal genetic variants are associated with direct cleavage and chaotic mosaicism in human embryos. So something that initially we could have believed to be associated maybe to abnormality of the centriole, we contribute the important part for establishing centrosome in the fertilized egg, and therefore a normal spindle by building up a bipolar aggregation of microtubules. In fact, it is a maternally derived problem. We have now a very relevant to this talk, new evidence of how fertilization occurs as far as the mitotic spindle is concerned. So the dogma that after pronuclear breakdown, a single mitotic spindle is formed in order to segregate chromatids has been challenged by this study by Reichman and colleagues a couple of years ago. So what they showed, in fact, in the mouse is that as soon as the pronuclei are dissolved and chromosomes are released, not 1, but 2 spindles are formed, which is incredible. And they act and they live for a few hours totally independently. They functionally independent. And only at a later stage, they fuse into a single segregation machinery. And that is followed by anaphase in the first cleavage division. So this is something entirely new, which actually can explain some of the disturbances of development we see in the human. If we experimentally in the same system increase PM breakdown, PM distance at breakdown, what we achieve is the formation of a tripolar spindle with the implication of a disrupted first cleavage, or a double spindle still existing as such until the time of cleavage, and therefore we have the astonishing finding of having the 2 parental chromosomal complements segregated independently, and that gives rise to the formation of binucleated 2-cell embryos and the mixing of those parental complements only at the 2-cell stage, not at the fertilization stage, which I found really amazing. Now, the important thing to notice here is that this is a single micrograph published in JAG, in fact, last year, suggests that something similar can occur in the human, as you can appreciate by 2 structures that can be interpreted by separate spindles at the first cleavage. Finally, I would like to discuss the topic of change of cell design at the transition between oocyte and zygote life. And that occurs as an effect of the formation of the 2 pronuclei that are congressed together at the very center of the fertilized egg. Details of this system, of this mechanism, are still poorly understood, but we know for sure that this centering of the pronuclei is very important. Occasionally, this newly acquired symmetric organization is lost as an effect of distancing of the pronuclei, or peripheral drifting, or even breakdown asynchrony. By these 3 modalities, maybe others, symmetry is lost. And I'm going to show you only one example of that. Usually we have the breakdown of female and male pronuclei very coordinated in time, with very minor negligible difference in time. And occasionally this can occur with some degree of asynchrony. It is a rare event, but if it is present, it gives rise to major disturbances of cleavage in terms of division of the fertilized egg in free blastomere or even cleavage arrest. And in this sequence, you can also appreciate how not only are pronuclei lost asynchrony, but the entire cortex has been upset by instability. So to conclude, and I hope I gave some contribution to the discussion, I hope I've been able to convey the message of the importance of cortical actin, which has relevance in terms of appreciation of significance of some forms of oocyte dysmorphism, but in a more physiological fashion, we have appreciate how polarization of actin, acquisition of differential cortical tension of the oocyte surface is fundamental for correct extrusion of polar body 1 and 2. We have also noticed how the integrity of the spindle is fundamental, as we have always learned, not only at the meiotic stage, but also at the later stage when the meiotic organization is converted in something slightly different. At the meiotic stage, we have the importance of segregation of chromatids, but we also have at the mitotic stage something even more profound, which is the support of a regular cell division as an effect of the presence of a bipolar spindle. Finally, I've tried to underline the importance of the acquisition and preservation of bipolar symmetry at the PN stage, which is something that usually is acquired, but is not necessarily for good. Sometimes towards the final stages of fertilization is lost, with very bad news for the ensuing embryo. And this leads to the final point, which is relevant to the mouse study I showed, that suggests that perhaps we should rewrite or reassess the crucial step of fertilization. And I think I finished with that. Thank you, Gary.
Thank you very much. That was a fabulous talk. Appreciate that very much.
Thank you.
I have, and I'm glad, as Gary pointed out, Giovanni is indeed an expert in cryobiology, and he has authored some fantastic work. And along those lines, let me just ask you, it strikes me in trying to put together David's presentation and yours,
Sure.
that the crucial steps that have occurred to lay down an appropriate actin cortical network in an appropriate manner that allows development. But in contemporary IVF, one of the things we now do is that subject that oocyte to an influx of cryoprotectants in the process of vitrification. Then upon warming, an efflux of that influx of more water, and then we press through the cortex a glass rod to fertilize it. So contemporary oocyte cryopreservation and warming strategies, some of your thoughts on that and how it's intervening in these appropriate mechanisms that you've described.
Yeah, well, if I go back to my colleagues' early study on oocyte cryopreservation, one of the findings we were able to observe was a partial loss of cortical actin integrity after slow freezing. And if you remember, those oocytes would survive nicely, at least in our hands. They would fertilize according to normal rates of development, but then they would have a very delayed development, sometimes with massive fragmentation, which is something that probably is reminiscent of damage to the cortex we were observing in confocal studies. Of course, with vitrification, the scenario has changed, but something that is suffered by the oocyte may be somehow regained, maybe somehow repaired, but everything comes to a cost. So when we compare, the same number of oocytes fresh cryopreserved, I'm not sure we're going to have there may be difference in terms of net generation of births.
I think, those are really, really important things for us to think about, right? And I think, I see a question here by Mitch talking about oocyte cryosurvival, but yet when you really ask the next question about developmental competence, yes, we're much better than we were a decade ago, but we don't have the answers. And I think it really comes to some of the things that David Albertini brought up, is that, there are cytoplasmic machinery, not just spindles and actin, but things that regulate that, right? Kinases and phosphatases that regulate metabolism, but also regulate spindle dynamics and things like that. And if those are compromised in any way during cryopreservation, you still have to repair, right? And it just kind of knocks down things a little bit every step.
And if I can say something more, I mean, we should not forget that we get excellent results with cryopreservation of eggs when we cryopreserve cells from good prognosis patients, okay?
Right.
Young patients and donors. But when you do the same exercise with, older age patients and patients with less favorable prognosis, the situation changes dramatically.
Right, and there is, and I think it's important that we don't, I don't think we fully recognize the amount of sublethal damage that happens with cryopreservation and how you have to repair those things. And those repair mechanisms require energy. And so all of those can actually affect later development.
In fact, I'd like to mention another study in which this is again a slow freezing, but this is very instructive of how things may happen during cryopreservation. When you cryopreserve oocyte from young and older patients, there is after slow freezing, a loss of spindle integrity. What happens is that the oocyte of younger patients are able to reconstitute a normal spindle, and they keep it as such over the next few hours after warming. But the same thing doesn't happen with the same efficacy in oocyte of older patients. So again, we are good at cryopreserving, but if oocytes are good. Otherwise, I'm not sure the cryopreservation is still optimized in our hands.
Well, that's an excellent point, and thank you very much. By the way, before we move on, and there's lots more questions that we will get to in our question and answer session, but let me give a quick plug. My very favorite paper, intellectual treatise, I think, of slow cooling, was authored by DeSantis and Coticchio, and it was in, I think, RBM Online. It's an outstanding paper, and if you want to really understand the ins and outs of slow cooling, I recommend that paper. Again, it's, I think, a classic. Well, in the interest of time, we will come back and have more questions, but I want to get to our third presentation, which we've been leading to as clinical embryologists, and that is, let me introduce Rebecca Krisher. Dr. Krisher is a graduate of Hanover College, which I believe is on the Ohio River. Is that right, in Indiana? Yeah, beautiful.
It is.
And from there, she's done quite a few things. She's been in research at Granada. She's been in clinical IVF, but she proceeded on and received her master's degree from North Carolina State University, and then went to onto her PhD work and received her PhD from Virginia Tech. And she is currently the research director for Colorado Center of Reproductive Medicine. And she gives a brilliant presentation on clinical IVM, a topic I think we've all anxiously been waiting to hear about. And Rebecca, thank you very much for participating. So here's Rebecca Krisher.
Thank you so much. I really appreciate the invitation to be here. It's my pleasure to talk about IVM. It's one of my passions, and I hope after this presentation, one of your passions as well. So I think this is a really great way to kind of bring everything we've talked about earlier today together and to think about how we can apply some of those principles in the clinic. So I'm going to start with this slide, and it was the cover story of the Eschera newsletter more than 4 years ago. It talked about the maturity of in vitro maturation, a shift at last from hype to hope. And while many of us were very excited and encouraged to see IVM kind of percolating up to the top, you know, maybe it is moving a little more slowly than we would like. So what I'm here today to talk about is, you know, what are the advantages and disadvantages of IVM, where does IVM fit in, and share some of our experiences with IVM as well. So I'd like to start with this question of where does IVM fit in? Certainly, I think in a historical sense, we thought about IVM as a way to reduce ovarian hyperstimulation syndrome. So with that controlled ovarian hyperstimulation that David talked about in the first talk, you know, we would often get negative side effects that really led to some severe health consequences for women that ended up with OHSS. So recently, I think, unfortunately for IVM, the advent of GnRH agonist triggers coupled with embryo vitrification and FETs has really reduced the incidence of OHSS in our patient population, which is great, but I think it kind of reduced the incentive for us to develop IVM as a clinical treatment. However, I would argue that outside of the OHSS advantage of IVM, that there are other advantages as well. And I think one of the biggest ones in my mind is the patient burden that could be reduced with the use of IVM. So with IVM, you have many fewer FSH injections, if any at all, and no trigger injections. So that's much less injections for the patient, which results in reduced drug cost and reduced monitoring costs as well. The time commitment that's required for fertility treatment is certainly decreased, just given the reduced stimulation. And this is really important for oncofertility patients, which I'm going to come back to at the end of this presentation. It can also be done with no stimulation at all, so it can be done really almost immediately for particularly important for oncofertility patients, but really applicable for other patients as well. And as I mentioned, less frequent monitoring. And so I think these recent reports that we've seen come out in the literature in just the last 2 or 3 years, we saw some really nice abstracts at ASRM last year about clinical IVM. And I think this has kind of renewed our interest in IVM, in addition to really understanding more about cytoplasmic maturation in the oocyte that David talked about, and thinking about how we can kind of combine this approach of in vitro maturation to maybe even potentially make a better oocyte, along with giving our patients more options. So when we think about patient selection, to me, at least where we stand right now in the clinical implementation of IVM, it's really important to select the right patients for success. And right now, that really means PCOS patients, primarily because they have a high antral follicle count. So you really need to start with more eggs to compensate for reduced egg maturity. So we don't get 100% of the eggs that we collect to mature in current IVM systems. So there is a little bit of a higher rate of attrition there than in a normal IVF cycle. So you need to start with a good number of eggs to really be successful. And that's why, unfortunately, at least not yet, it's really not a good treatment for patients that are looking for a last resort. So patients that have DOR, POI, advanced maternal age, who have failed cycles, or who have been recommended for egg donation. Now, we have put some of these patients through IVM. They typically are not very successful. And so, as you'll see later, really the focus on PCOS and high intrafollicle count patients, at least as we are right now, where we are right now, is the way to go. Now, I think, as we learn more about clinical application of IVM, we get better at IVM, there may be opportunities to treat some of these other types of patients, but not yet. So I want to, before we talk too much further, define IVM. And it's probably, you think that's maybe a silly thing to do because IVM is in vitro maturation. But in fact, we talk about IVM in a lot of different ways. So in the literature, it's kind of difficult because there are papers that talk about IVM using a trigger shot or not using a trigger shot or using gonadotropins or no gonadotropins. So it's difficult to compare between different protocols and different approaches because they're all called IVM, even though they are in reality very different things. And so what I would like to just point out is that there really are two main types of IVM that you can kind of break into different barrels. So the first is trigger IVM, which I think more people have done. This is also called truncated IVF. And this is where HCG, or a trigger shot, is given prior to retrieval of the oocytes, but you're retrieving oocytes from typically smaller follicles. 12 millimeters or less is pretty common. So because of that HCG injection, meiotic resumption has been initiated. And so you will almost always retrieve some mature oocytes along with some immature oocytes. Typically in those cycles, the mature oocytes are the ones that make blastocysts, are chosen for transfer, and actually produce the pregnancy. So it's a little difficult to really understand a lot about true IVM from those trigger IVM or truncated IVF cases. So what we really think of as true IVM is the original definition. And that was made by Edwards in 1962, so many years ago, but I think it still holds true. And that's the maturation in vitro of immature cumulus oocyte complexes collected from antral follicles. And so you may hear this called no trigger IVM, pure IVM, true IVM, but the bottom line is that no HCG trigger is given. So that trigger for meiotic resumption has not been seen by the oocytes. And so as a result, you're only collecting immature oocytes. No mature oocytes are recovered. It's also important to understand that this does not include those GV oocytes that are recovered in a typical IVF cycle after the trigger shot that we often put through IVM and call rescue ICSI or rescue IVM. That's not what we're calling pure or true IVM. So the other thing that it's important to keep in mind here is that either of these approaches, IVM with or without a trigger, can be used with or without gonadotropin treatment. So just keep those things in mind as we move forward and talk about the literature. And so I wanted to just give you a pretty brief overview of what is out there in the literature in respect to IVM and clinical use. One of the more recent and larger studies was a retrospective study out of Vietnam with 919 women. But the interesting thing here is this is not true IVM, this was trigger IVM. So both IVF and IVM received gonadotropin and received a stimulation or an ovulation trigger shot. The difference being that the IVM oocytes were, of course, recovered at a smaller follicle size. And so they asked the question, what's the chance of live birth after one IVM cycle with a trigger or an IVF cycle? And what they showed is that the number of mature oocytes, embryos, good quality embryos, and frozen embryos were all significantly lower after IVM cycles. The cumulative live birth rates were not significantly different, although they were a little lower with IVM at 39% versus IVF at almost 50%. They did see a reduction in hyperstimulation syndrome, which you would expect. And they concluded that in women with a high antral follicle count, IVM is an effective ART option. And although in this study not as good as IVF, I think it does show that use of IVM can be an option for some patients. But again, this was with the HCG trigger. So what about when we look at cases with no trigger? So only FSH. And in this particular study, well, all of these studies, they did not use an HCG trigger. And most of these studies came out of the lab in Brussels that's headed by Michelle DeVos. So in 2011, so really quite a few years ago now, they started looking at IVM as a potential clinical treatment. Their first prospective study was published in 2011 with 39 patients and 73 cycles and a 32% pregnancy rate per embryo transfer. In 2012, a slightly larger study, 121 patients with 239 IVM cycles, again, no HCG trigger, and 35% pregnancy rate per FET. The last study that I want to mention today came out in 2013. And again, no trigger IVM, and they showed a 16% live birth rate per ET, a cumulative live birth rate per patient of 22%. And this was 79 patients. So again, some early indication that IVM could be successful. I'm going to go back just a slide here. I wanted to point out that in this last study in 2013, the live birth rate per retrieved immature oocyte was only 1.1%. So I want to compare that to what David talked about earlier, where if you look at a regular IVF cycle, that comes out somewhere around 5%, depending on your patient population. So IVM right now, at least in 2013, is a bit lower than that. And I think that really emphasizes some of the inefficiencies that still remain in IVM. I think this has probably improved since that time, at least in our hands. But it does, I think, point out some of the attrition that you see in IVM. So what I want to talk about now is the idea of pre-IVM. And I think this is really where we tie together some of those important cytoplasmic components that David talked about early on in this presentation. So pre-IVM simply means that you are aspirating an immature oocyte and holding it in that immature state in the laboratory for some amount of time before you then put it into an in vitro maturation media and release meiotic arrest and progress to metaphase 2. So the idea here is that you're able to support the oocyte in vitro in the germinal vesicle stage and allow that cytoplasmic maturation to happen. And then once that process has hopefully been completed, you're able to release meiotic arrest, progress to metaphase 2, and you have a higher quality oocyte as a result. So there's been several groups around the world working on this protocol, and these are some of the first 2 papers that have come out, again, from the group in Brussels. And in 2017, they reported where they were giving only 3 to 5 days of FSH with the largest follicle not greater than 10 millimeters at retrieval. They collected these into a media with IBMX, and not to go into too much detail, but IBMX and then C-type natriuretic peptide both work in different ways to maintain cyclic AMP and cyclic GMP at high levels within the oocyte. And this is the natural means of keeping the egg in germinal vesicle stage. So you have to keep that cyclic AMP and cyclic GMP high. Once they fall, then the egg will progress to metaphase 2. So what they did here is they collected an IBMX. They had the pre-IVM period for 24 hours with C-type natriuretic peptide. You might also know this as CNP or NPPC. And then they followed that with 30 hours of IVM in a media with FSH and amphoregulin. And what they saw was that using this pre-IVM system, they were able to improve oocyte maturation, improve the resulting embryo quality in the pre-IVM system versus the IVM system alone. In 2019, they published another paper. This was really a basic research paper looking at this 2-step IVM system, which they call Kappa IVM. Again, collecting an IBMX, again, the 24-hour pre-IVM period with CNP and the 30-hour IVM period. And they, in this study, looked at gene expression in the blastocyst and imprinted DNA methylation in the blastocyst that were produced in this two-step IVM system with the pre-IVM period versus controlled ovarian stimulation in a normal IVF cycle. And what they saw was no differences. So the pre-IVM IVM period does not, at least according to this small study, seem to impact methylation or gene expression in the blastocyst. And this brings up a really important point, and that is safety. We don't know a lot about the safety of IVM. You know, studies are coming out all the time, but because IVM has not been widely used, there's really very little information out there about it. So I did want to point out some studies that have looked at this question, seen no increase in imprinting errors, no increase in aneuploidy of the day 3 embryos, no differences in cellular morphology at the electron microscopy level between IVM matured oocytes and in vivo matured oocytes. There's been a couple of studies as well, follow-up of IVM children, no difference in mental development at 2 years of age, and no adverse effects on neonatal health. So these are all fairly small studies. There's certainly more work to be done here, but initial studies are encouraging. And so I want to spend a few minutes here just talking about our experience with IVM. We started about 4 years ago down this path, and we started our first IVM clinical trial based upon some work that we had done in both the mouse model and the cow model. And we had developed, similar to the group in Brussels, a way to maintain using cyclic, NPPC or CNP, maintain those cyclic AMP, cyclic GMP levels high in the oocyte and maintain meiotic arrest. So what we did is we started with a variable stimulation period based on the patient response. So ended up being usually around 5 days, but anywhere from 3 to 10 days of FSH at a lower dose of 150 IU. And then we targeted retrieval when the largest follicle size was about 14 millimeters. We had a pre-IVM period of just 2 hours. If you think about the size of the follicle and the pre-IVM period kind of as a teeter-totter balance, the larger the follicle, the shorter the pre-IVM time needs to be, because that egg has had a chance to grow in the follicle and develop some of those cytoplasmic components already. Whereas if you're working with a very small follicle, you may need a longer pre-IVM period for some of those processes to take place. So in this, our first trial, we just had a short 2-hour pre-IVM because we had a slightly larger 14 millimeter follicle. And then followed by an IVM period of 24 hours. And I'm just going to point out just a few results from this study. We don't need to go all the way through this table. We got about 9.5 eggs per retrieval, only a 43% maturation rate to M2. So this was a little disappointing. We certainly had hoped for better. And I think you'll see that we have improved that particular point. But it is one of the places where you do get a lot of attrition in IVM. 67% fertilization, 40% good quality blastocyst. And interestingly, what we saw is that 75% of the blastocysts that were produced from IVM eggs were euploid. And this was a little bit higher than what we see typically for patients that were age matched to these 11 patients in this first study. So we felt like we were maybe getting a better quality egg. We were seeing less aneuploidy in some of these blastocysts. And of these 11 patients, we had 6 of them make embryos. 5 of them had FETs. Unfortunately, 1 patient did not have normal embryos. But of those 5, 4 resulted in positive pregnancy tests. We had no losses and 4 live births. So we were pretty excited about this initial data with this first cohort of only 11 patients. But what's really interesting, these 11 patients had multiple different reasons for their infertility. However, of all the patients that had live births, none of them had previously had a recommendation for donor egg. So I think, again, this just shows how important it is to choose the patients in your study. Our second clinical trial is ongoing currently. We did tighten up our patient population here, so only PCOS patients that were 35 and younger, an antral follicle count of 25 or higher, and a BMI of less than 30. So in this protocol, we were going for a slightly smaller size follicle at 10 millimeters. And so what we did here is we had 4 days in every patient of 150 IU's FSH, so only 4 injections. Then retrieval, we came pretty close to this. We ended up at an average of about 11. But then we recovered those oocytes into the pre-IVM media and cultured in pre-IVM for 20 hours because we had, again, a smaller follicle size in this trial. Then IVM media for 27 hours, and then we removed the cumulus, looked for maturation, and followed by ICSI, culture, PGTA, and FET. So again, lots of numbers here. I think I'm just going to point out a few of the important ones. As I mentioned, our largest follicle on the day of retrieval averaged about 11. We recovered an average of 25 eggs per patient. Eggs matured at 30 hours, average 12 per patient, and total matured eggs 16 per patient. A 57% fertilization rate, only 7% day 5 good quality blastocyst, 24% total good quality blastocyst. So while we got a little bit better at maturation, we did suffer a bit as far as our blastocyst development, and most likely because we were working with smaller follicles in this study, but some work to do there to be sure. Again, a good percentage of those blastocysts were normal. And of the 14 patients that have gone through this particular protocol so far, 10 of them produced good quality blastocysts. All of them had at least one euploid blastocyst. And we've had 7 patients who have undergone embryo transfer to date. 4 of them are pregnant. We've had 3 healthy babies and 1 ongoing pregnancy. So before I finish, I want to talk just a bit about fertility preservation and the use of IVM in that arena. So I think it's pretty well known that more young women are surviving their cancer treatment. So it's really become an important part of the treatment of their cancer to consider preserving their fertility. And there are several approaches to doing this. Most efficient and probably the most common is to undergo ovarian stimulation, basically an IVF cycle, retrieve eggs, and either vitrify mature eggs or if they have a partner to fertilize those eggs and freeze the zygotes or the blastocyst for future use. But there are some cases where ovarian stimulation is contraindicated, so it would not only stimulate their ovaries, but it would stimulate their cancer as well. And so in this situation, ovarian tissue is often harvested and vitrified for later use after their cancer has been cured. But I think it's really interesting to consider IVM as having a place in this strategy as well. So this was a paper that was published earlier this year, just in February, and it's a case report demonstrating the first birth that was achieved after using in vitro maturation for fertility preservation in a woman with breast cancer. It was led by a group out of Paris by Michael Grinberg, and he described a 29-year-old woman who had breast cancer that was estrogen and progesterone receptor positive. So they could not do an ovarian stimulation. The chemotherapy was already scheduled, so the patient was offered oocyte vitrification after IVM without ovarian stimulation. They did an ultrasound. They found 17 small antral follicles on her ovaries at cycle day 16, and they actually did the retrieval shortly after on cycle day 22 and retrieved 7 immature oocytes, of which 6 matured in vitro and were vitrified. So 5 years later, those oocytes were thawed, underwent ICSI, 5 were fertilized normally, and 1 cleavage stage embryo was transferred and produced a healthy term baby boy. So I think this is a great example of how IVM could be a viable and an efficient option for female fertility preservation, particularly for those patients that really can't undergo hyperstimulation or ovarian stimulation. The second paper that I just wanted to point out was published just last month in August of 2020. This was a group led by Michael DeVos out of Brussels again. And what they did is they combined the ovarian tissue cryopreservation with in vitro maturation. And so as you're processing the ovarian tissue into small pieces, you actually release some oocytes out of those small antral follicles on the ovarian cortex. And so what they did is they collected those oocytes and matured them in vitro. So they combined those two techniques. They reported on 77 patients. They collected over 1,000 COCs. Only 39% reached metaphase 2. Again, we're talking about oocytes from extremely small follicles here, but they were able to mature some oocytes, 64 patients vitrified oocytes, an additional 13 patients vitrified embryos after ICSI. And so 2 of the patients with vitrified oocytes have had those warmed, and 1 of those 2 has delivered a healthy live birth. There were 5 patients out of the 13 with vitrified embryos who have warmed them, and that has resulted in 2 healthy babies. So that's a live birth rate per patient of 43%. And so they argue, and I would agree, that in vitro maturation really does add value to ovarian tissue cryopreservation as a means for fertility preservation. So I just want to end by summarizing some of the large themes that we've talked about. And I hope that you will agree that IVM really could be a useful alternative approach, not only for oncofertility, but also for some of our PCOS patients. Certainly, it really takes ovarian hyperstimulation out of the question. You know, that's really, I think, very well recognized. But I think some of the other advantages, like reducing drug cost and minimizing patient burden, are really underappreciated. So far, what we've seen is that when we get to the cleavage or blastocyst stage and transfer those IVM-produced embryos, that the implantation rate is similar to that of IVF blastocyst. And one thing I think to think about is that there is a high rate of attrition, but one thing that means for us is that fewer excess embryos are produced. And that's a little counterintuitive because we always like to have a lot of oocytes and a lot of embryos. But as many of us know, we have a lot of embryos stored in cryotanks around the world, and patients struggle with how to deal with those embryos. So if we can produce fewer embryos because we're dealing with a higher quality oocyte, that makes a pregnancy so we don't need so many embryos, that may be a real advantage. Certainly encouraging effectiveness and safety data is emerging more and more all the time. And as I've talked about, it could be very useful for oncofertility. There are some disadvantages, of course. You know, it's not a perfect technology yet, but I think it's well on its way to becoming applicable. But one of the main drawbacks right now is that it's not applicable to all of our patients. So we're really talking about, at this point, patients with PCOS and a high antral follicle count. In addition, patients with a very high BMI over 30 also may be problematic because the retrieval is a little bit tricky. You're trying to get cumulus oocyte complexes that are more tightly adhered to that follicle wall. Takes a bit of twisting and scraping of the needle, so you really need to have good access to those ovaries. Certainly, it seems so far that there is a lower efficiency of getting to metaphase 2, of getting to the blastocyst stage, but those blastocysts do seem to be of very good quality. We need more large clinical trials comparing true IVM without a trigger to standard IVF, and we just don't have those now. I think they're coming, but they're not quite there yet. And then, of course, we need long-term safety follow-up. So I think what this really means for us is that, you know, we can do IVM clinically and be successful right now. I think as we do more, as we learn more about the human oocyte and about how to support that cytoplasmic maturation, that we're going to get better at doing this, and we may be able to branch out to some of those different types of patients. The other thing that I find really attractive is that pre-IVM period. So I think what that pre-IVM period will allow us to do eventually is to support different types of oocytes in different ways. So for example, if we know that oocytes from women of advanced maternal age, say, have mitochondrial deficiencies. Maybe we could add something into that pre-IVM media and support those deficiencies in such a way that we significantly improve the quality of their oocytes. Again, we're not there yet. We still have a lot to learn, but I think this pre-IVM period really opens the door to taking advantage of some of that cytoplasmic component processes that David talked about that we're going to be able to better manipulate in the future. So with that, I'll stop, and thanks so much for all of your attention. Thanks for having me.
Great. Thank you, Rebecca. This is Gary. I guess I'll kind of start with the first question. I'm going to try to meld this, a couple questions that I've seen come through, and maybe have you address it first, but I think also David and Giovanni can address this too. It kind of gets back to, I mean, you just ended talking about pre-IVM and, you know, talking about a few hours, maybe 6 hours. David talking about COS and, you know, trying to make a mono-ovulatory to a multi-ovulatory type of a model or animal per se. I have to say, going back years, one of the things that discouraged me was thinking about, you know, what folliculogenesis and oogenesis is in vivo, in situ, and how do we really think. I always thought maybe we're being a little naive to think that we can just pluck it out and put it in a petri dish and get it to go to metaphase 2 and kick out a polar body like David said, but that doesn't tell us everything. Now maybe thinking about, you know, pre-IVM with a couple hours of IVMX or cyclic, you know, an inhibitor of nuclear maturation. Mm-hmm. But does that really move forward? And it gets to the other question here I had from somebody asking about, you know, during folliculogenesis, and David brought this up, we have endocrine and paracrine, right? And so when we pull an oocyte out of a follicle, independent of what antral size, you know, what are those paracrine things that we're missing? And, you know, one would love to have that magic wand to know what it is, but kind of give us some insight, because I don't know, you know, if IVMX is that magic pill that's gonna help us get this through.
Right, and I think just to comment on IVMX really quickly, it's not something that we use here, and I think, you know, in general, we're probably moving away from more pharmaceutical ways and thinking about more physiological ways of maintaining that arrest, such as the CNP molecule. But I think in general, I would point out two things. One is, I don't think we have to completely recapitulate the time scale of oocyte maturation in vitro compared to in vivo. So it may take quite a long time in vivo. It doesn't necessarily have to take that long in vitro. I don't think we know that for sure, but that's my suspicion, is that some of these processes can be completed in a shorter amount of time in vitro if we give the oocyte the opportunity to do that while maintaining meiotic arrest. The other thing I would say is that just, you know, in the past 5 to 10 years, we know so much more about the mechanisms of meiotic maturation and the communication between the cells of the follicle and the oocyte that we can recapitulate some of that in vitro. And I think that has really, to me, been the key in moving this forward, because we can add some of those things from the follicle, some of the things we know should be there in that communication into the petri dish in the pre-IVM media or the IVM media both. And I think that will allow us really to support that oocyte in a way that we just haven't been able to do before.
David, you want to add something about that with regards to follicular development?
Okay, yeah, I'd like to follow up on that. And I think two things. First, pre-IVM conditions have evolved over, I would say, the last decade, from the days when we all just used IVMX to hold the COCs until we washed them out, okay? And, you know, Rebecca's right. I give credit to, you know, really quite a few people for understanding what the real signaling mechanism was for maintaining meiotic arrest. okay? Way back when, we just thought everything had to uncouple and the oocyte was programmed to go forward. And it was programmed to go forward, but only supported nuclear maturation was
Right.
really what Epping and these people found out back then, okay? So when you bring it back to a COC, it turned out that the CMP, the fact that this is not an LH-mediated process was probably one of the biggest surprises, okay? I mean, 20 years ago, the LH SIRS caused uncoupling of gap junctions and you're off to the races, okay? It ain't that simple anymore. It just isn't. There are all these not only intrafollicular factors like CNP, okay, that essentially have been shown. I don't know, how closely, Rebecca, you've looked at this, but if you talk to Rob Gilchrist or the Australia gang, this is happening because you maintain the TZPs, which we could never do before. As soon as we took a COC out, those cells retracted, they changed their polarity, and that's what let the oocyte go off its merry way. So now these holding conditions are actually preserving an architecture that was set up not just to maintain a rest, but to prevent those cells from luteinizing. So let me try and tie this to the clinic. We are subjecting patients to high levels of gonadotropins. We love to drive them with as much FSH as we can, okay? And I think it was 2004 when we published studies with knockout mice that we showed, what the direct effect of FSH is on a developing follicle? Retraction of the TZPs. It's retraction of the TZPs. So I would argue that, yeah, we can pull all these oocytes out, okay? A lot of them mature. With ICSI, of course, we bypass any intrinsic differences, okay? We don't know who's real. We make lots of blastocysts, but at the end of the day, we are collecting a cohort of oocytes that are so heterogeneous, okay, that it's very difficult to know what they need. And now I'll just close with Rebecca's point. Yeah, the pre-IVM, this holding state, would give us a chance to tailor a therapy, if you will, on a per oocyte basis, depending on the age of the woman it came from, depending on whether, these came out, if they come out of a PCOS patient, then you got other issues going on in terms of androgens, okay? So I share your enthusiasm, Rebecca. I think we're at a turning point because finally there's some real science that we can put into a clinical context, which we didn't have before.
It's an exciting time. I think there's lots of opportunity, and certainly we're in early days and we have to get better at just IVM in general. But I think that the possibilities that IVM might offer us are really exciting.
I think that I have been thinking and discussed with Michelle Lane before her untimely passing the idea of precision embryology, because I think there is some therapeutic culture in the horizon that we could all use. But precision pre-IVM is exciting to think about as well. That's a great concept. I think Jacques Cohen, to kind of change gears early on, asked, I think, a really intriguing question. And that is there any work being done on studying the maturation of the oolemma during maturation? We do see in clinical in vitro fertilization activities quite a wide variety of behavior of the oolemma. As he points out, if you try to place a needle into a germinal vesicle oocyte, it's often not very successful. It can be later on, as we do in ICSI, but we do see that variety in membrane quality. What's being done about membrane studies in maturation?
Well, what we experience when we do ICSI and we see all of these different degrees of resistance, even within a patient, okay, it's not really an OLMO problem, okay? This is about the mechanical tension that's created when you build that cortical net, okay? Which, and that's, I think that's the nature of the beast now, is we need to understand more about how that network is assembled and stabilized. And there's been a beautiful series of papers to essentially define what this is first at the animal mammalian egg level, and now more recently at the human level. And it's called the subcortical maternal complex, the SCMC. And I think that's where the action is going to be in terms of Jacques' question. Okay. This is basically a device that's assembled at the time of maturation, and it's used to store proteins and messenger RNAs of maternal origin that do not get used until the time of compaction. And you can imagine, for example, with all the fragmentation that we see anyway, that this is either a reflection of defective assembly of the SCMC, or more likely is a way that embryos are discarding important determinants for example, remethylation of the genome. Because the dimethyltransferases, at least one of the important ones, are actually located within this. That's my 2 cents worth, but I think it's another one of these things that as we explore the IVM paradigm, we'll actually get a chance to study it in more detail.
And I think that's a great lead into something that you said earlier, David, about, we're really looking for those not single biomarkers, but the multi-parametric feedback that we can get that will tell us about developmental competence. Because, I think, there's no doubt that there are these things happening in reconfiguration that we don't see at a light microscope level in the cortex, let's say, what we're talking about here. The other one, some of the early work by DeSantos and colleagues looking at the pulse scope and, how you think it's a metaphase 2 oocyte, but it really is not a metaphase 2 oocyte. And, we and others have shown that energetic enzymes, kinases that regulate energetics, are localized at those places where you have that segregation. And if they're not functioning correctly, that in itself looks like a metaphase 2 oocyte, but it's not a metaphase 2 oocyte. So, I think, we're going to need to step beyond visual observations with good solid data to tell us, not just this one thing tells us which oocyte is going to be developmentally competent, but the multiple things that will feed into it. And so, I think, that absolutely the modifications of the sub-olemal region, is likely something we haven't studied well enough and is part of cytoplasmic maturation.
Yeah.
Great.
Excellent. Rebecca, one quick question, and that is, I know it's going to pop up, and that is the availability of medium for doing pre-IVM and IVM. Are there appropriate media available, or is that something for the future? Commercially available?
Yeah, unfortunately not really commercially available right now. I'm hoping that it will become available in the future. Right now, people that are taking the pre-IVM approach are making media in-house, as are we. And it's difficult because there are a lot of really specific peptides that you need to add into that mix. And, I think the hurdle really is getting, governmental approval to add some of these things to human eggs that really haven't been used on a large scale before. So it is an approval hurdle, to be sure. I think, until that time, hopefully collaborations will pop up that, we can share media between groups that are interested in IVM. I think there's a lot of IVM kind of supporters that would love to see IVM be tried more, on a larger scale. So, yeah, but it's a problem, to be sure.
Yeah, got it. So I think if there are no other pressing immediate questions, I'd like to first of all thank both David and Gio and Rebecca for a fantastic presentation, and Gary for your excellence in co-moderating. Again, I want to thank the IVF Initiative platform for providing this mechanism for us to all have these discussions, and remind you again, if you want to go onto the website and look up this session, all of the speakers' biographical sketches are available to you. You can learn more about them and follow their work. Also, just a reminder, next week there will be session 33 on sperm function, and be sure to get onto the website and register for that. And I think with that, unless there's anything else we need to do, again, I want to thank the people behind the scenes that have helped us produce this. And absolutely, again, Gio, thank you. David, thank you. And Rebecca, thank you so much on behalf of both Gary and me. Thanks so much.
Very much.
Thanks, everyone.
Okay.
Bye now. Stay